What's up, Mr. Chang? You're at work? Nice. Don't let me distract you. I don't want to be that kind of an influence. Thanks, man. Have a good one. Don't work too hard. Nice. I want to get a little more info on these phase 3 COVID vaccines. I've been a little slacking on my keeping up to date on what exactly is going on with these vaccines and the differences between them. So I'm going to do a little bit of research today, educate myself, get myself up to date, make sure things are looking. You never want to be like completely out of the loop on some of these things. So let's do a little bit of this. Like, let's look up this one. Let's look up this one. Look up this one. And see what they're about. Which ones are mRNA vaccines? Which ones are attenuated vaccines? All right, here we go. New Scientist, New York Times, CNN, NBC, KNN, WC, Wall Street Journal. Let's do exciting. I'm telling you. Exciting, right? Everything you need to know about the Pfizer COVID-19 vaccine. December 3rd. All right, this is pretty new. Okay. How effective is this vaccine? 95%. The phase 3 trials of the Pfizer vaccine involve 42,000 people. What's up, Alexi? So you got 42,000 people. Half got experimental, half got a placebo. So 170 people fell ill with COVID-19. Only 8 of them were in the vaccine group. 162 had received the placebo. Okay, so that's pretty good. That's pretty good right off the bat, right? So around 5% of the cases were in the vaccine group, which was where the 95% figure comes from. That is a very healthy number. The World Health Organization had said it would be happy with a 50%. Whoa, low standards, World Health Organization. So basically, this is pretty good, right? So of the 170 people that fell ill, only 8 of them. So that's 8 out of 170, right? So that's where the 5% came from. Okay. What is this one? Oh, this is the mRNA one. So the Pfizer one is the mRNA vaccine. So the active ingredient is a messenger RNA. It carries instructions for making the virus's spike protein, which is used to gain entry to cells. The mRNA is synthetic, not extracted from actual viruses. Okay, so they made the mRNA. So that means they must have sequenced the protein, figured out the sequence of amino acids in the protein chain, and then created an mRNA molecule that codes for that sequence of amino acids. So they kind of like back engineered it or reverse engineered it. It is delivered in a tiny sphere of inert fatty material called a lipid nanoparticle. Okay, all that we knew. The RNA-bearing nanoparticles are suspended in a saline solution and injected into muscle tissue in the upper arm. The mRNA is then taken up by the specialist immune cells, which follow its instructions to make the spike protein, just as they would do if they had become infected with the actual virus. The spike protein is recognized as foreign by the immune system, which mounts an attack against it. Antibodies, B and T cells, are activated, according to Ugur Sahin, the chief executive of the small German company BioNTech, which co-developed the vaccine with the U.S. drug giant Pfizer. An immune memory is also laid down, he says, which means the immune system has learned how to defeat the pathogen and is primed to mount a swift response if it encounters the coronavirus again. How long does the immune memory last? It's hard to say at this point because clinical trials weren't set up to answer that question, right? Because they're just trying to see if it was effective, not like its longevity. Worst case scenario, if it doesn't last that long, you could just get a booster anyway. So like, the important part is like using it as an immediate way to just sort of douse this whole virus. They only began dispensing second doses 4 months ago. The WHO says that a minimum of 6 months would be acceptable. Okay. It will become clear as time marches on and the volunteers continue to be monitored. Sahin says that he expects protection to last months or even years. Given what we know about natural immunity, that looks about right, says Eleanor O'Reilly at the University of Edinburgh in UK. She envisions people needing annual boosters at worst. So basically like a flu. No different than the flu at that point. How long will it take for immunity to develop fully after vaccination? The trial began assessing immunity 7 days after the second shot. We know that protective immunity builds up within 4 weeks of the first dose, but Sahin says that it appears to develop earlier than that. Further details we published. What happens to the mRNA in the body? It is active for, then it decays. We knew that. It's a 2-shot vaccine, so what happens if people miss their second shot is a single shot protective. 2 shots are needed. The second shot is required to attain immunity. The gap between doses in the trial ranges 19 to 42 days. Only 2% of people in the trial that missed their second dose. Missed their second dose. So it isn't clear what happened under those circumstances. Okay. Side effects, sometimes, but they're mild. In the trial, the vaccine was generally well tolerated, and an independent data monitoring committee reported. So let's do this actually real quick. All right, so first we've got Pfizer's mRNA vaccine. I'm just going to jot some notes down here just so I can keep things organized. Pfizer's mRNA vaccine requires 2 shots, is 95% effective based off on, what was it, N equals, what was the sample size? 42 grand, okay. Well, actually, no, it wasn't 42, it was actually only 170. That were affected, so this is 170. Requires 2 shots, is 95% effective based off of an N equals 70. Not sure. So we're not sure how long the immunity lasts. At worst, maybe a yearly booster. Guten Tag. Guten Tag, Fraulein. Matthias. I don't know what's the German version of Matthew. Matthias. No, German. Where is it? Matthias. How goes it, my dude? Side effects. Mild so far. What did we see? The worst side effect was fatigue and a headache. Only reported in, what was it? 2 to 4% of people. Got a lot of work? What are you working on? Join me. I'm working as well, basically doing a little research right now. Trial participants were aged up to 85. A tiny bit lower than the overall number, but still protective, and a much higher than some vaccine experts feared. The vaccine hasn't been tested. Okay, works on old people. Works, oops. Math and later Chinese. Nice. What's up, Nathan? Yep, I'm doing a little more research. I'm trying to put together some notes on the right now what I'm doing is I went to the CDC website and they show you the 5 companies COVID vaccines that are in phase 3 right now, large scale phase 3. So I'm just doing brief research on each of the 5, just so I can kind of wrap my head around the differences between them. So I started with I'm going backwards here. I started with Pfizer's, so that's what I'm looking at right now with this article. And just as a note, yesterday we talked about mRNA vaccines. I did a little research on that, so I want to make a note there. Let's see. One of your friends was asking how sister chromatids and homologous chromosomes are different since they look the same. That's a really good question, and that's like one of the hardest parts of learning about meiosis in the chromosome situation. So let's see, how could I explain it? So basically, and don't worry, we'll go over this. I'm going to spend some time in class talking about this on Monday and Tuesday. So firstly, like if you imagine, let me pull up a little bit. How about we pull up Paint here? So let's imagine we've got, let's say, here's our cell. Okay, and inside of our cell, we've got our chromosome. So let's say we've got, let's use red and blue. So what I'm going to do is red equals your maternal chromosome, the one you got from your mom. And we'll use blue is going to be your paternal. Okay, so in your cell, you always have one of each, right? So let's just say we're looking at chromosome right now. All we care about is chromosome. By the way, sometimes chromosome is abbreviated like exome. We're looking at chromosome number 1. So if it's chromosome number 1, I'm going to have a chromosome from my dad, my biological father. and a chromosome from my biological mother of chromosome number 1. This is a homologous pair. Okay? So what a homologous pair is basically the same chromosome number 1, that includes both the mother and the father's version that you received. Okay, so this is a homologous pair. And a homologous pair has a set of sister chromatids. Yeah, so in order for sister chromatids to form, remember, before what happens, I'm going to move my cam up here real quick. So what happens is, then this goes through S phase, aka DNA replication. So the DNA is copied. And now, what happens is each one of these is copied. So what you get is, now you get your father's chromosome is doubled, right? So now there's 2 copies of your dad's. And the same thing, there's two copies of your mom's, the maternal chromosome. So this and this are sister chromatids. Because they're exact copies of the original mother/father. I'll move my cam out of the way again. So those are sister chromatids. This whole thing is called these are still homologous pairs. Does that make sense? They're not identical. So sister chromatids are identical. So and this is where it gets confusing. And this is why a lot of students struggle with meiosis, because they get all confused about what's identical, when there's a difference between the word identical and the word homologous. So identical means they're exactly the same, like down to the exact like sequence of DNA on each chromosome. So sister chromatids are identical. Let's see, how can I do this? Let's do purple here. I'm going to circle this. Identical. Homologous pairs, not identical. Because remember, the mother and the father chromosome that you receive have different alleles. So for example, let's just take an example here. So on chromosome number 1, there's this gene for, let's say, eye color. So we'll say this is gene for eye color. I might have a brown allele from my mother and a blue allele from brown allele from my father and a blue allele from my mother. So notice that they have the same gene, but they have different versions of the gene, different alleles. So they're not identical, because my father gave me a dominant allele and my mother gave me a recessive. But of course, when they're copied during S phase, now of course, these are going to be identical. This is going to be a big B, and this is a big B. But on the mothers, they're also identical. It's going to be a little b, and that's going to be a little b. So you'll notice that the sister chromatids are identical to each other, but the homologous pairs are not identical. They're homologous, they're similar because they have the same genes for the same traits, but they're slightly different because they would have different alleles, potentially. Is that clarifying at all? So the alleles are together while homologous are dominant recessive. I don't know if I like the way you phrase that. I'm not sure if that's exactly, I don't know what you mean by that, but if I think it's what you mean, I don't know if I'm 100% a fan of how you phrase it, because I don't know if it's exactly right. The way to think about it is homologous pairs have different alleles, right? Like the dominant and recessive alleles potentially. Whereas sister chromatids have the exact same allele because it was a copy, right? And I think that's kind of the best way to explain it. Okay, yeah, no problem. Let me know if you need anything else explained. I'll just minimize this paint drawing and I can bring it up if you need help going over any part of it. So let's see. mRNA is advantageous. As a vaccine because it is, from what we learned last time, it is not subject to MHC restriction. And it does not require any actual viral particles to be injected. Does it stop people from catching and transmitting the virus? We still don't know. The trial was designated to test for symptomatic COVID-19 and confirmed infection with the virus. Assessing whether the vaccine prevents transmission, which is probably a prerequisite for attaining vaccine-induced herd immunity, is much harder. Pfizer says it is carrying out more studies on this important question. Okay. Some vaccines can paradoxically make a disease worse through a process called antibody-enhanced disease. Is that a risk? Yes, theoretically, but it hasn't been seen with this vaccine or against. So maybe sort of like something at the beginning. So vaccines have been able to be pushed into phase 3 clinical trials quickly, approximately 8 months, because due to the global pandemic, they have been issued a, temporary authorization for emergency use, meaning they have an expedited path to distribution that a normal non-emergency vaccine would not. Thus, why they are out so fast. Normal vaccines is on the scale of years normally to get approved. How does mRNA vaccine work? So normal vaccine works. mRNA vaccines work like this. So normally, remember in class we were talking about how antibodies worked last week? And I had talked about this idea that antibodies that you produce in your body respond to a specific antigen and like attack it. And that's how it recognizes like foreign things in the body. So if you get infected with COVID-19, your immune system will identify that virus as a foreign thing, and it will identify antigens on the virus and attack it. So normally when you get a vaccine, the goal is to teach your body what the antigens are so that they can produce antibodies against it, so that when you actually get infected, it can just kill the virus before it even makes you sick. Like it's just already ready for it. That's the way vaccines work. And so typically the way in the past, there's never been, I do not believe there has been a single mRNA vaccine for anything. I think that's true. Wait, is there any mRNA vaccines FDA approved? Let's see. Let's see. Oh, hey guys. No worries. Let's see. Is there any mRNA vaccines approved currently? I think this might be the first. Everything's all COVID stuff. I'm pretty sure there's no mRNA vaccines. But anyway, so essentially the way you can think about a normal vaccine would be you kind of like dose the person with like some virus that you've killed already. Like you either, you like treat it with heat so it kills the virus, so it's not like active, so it can't make you sick, but the antigens are still there. So it's like you're injecting like all of the antigens into the person, and your immune system can react to it without the actual like lethalness or deadliness or, activity of the virus. With the mRNA virus or mRNA vaccine, instead of having to inject pieces of the actual virus or like dead virus, what they do is they basically figure out how to make a sequence of nucleotides, which is mRNA, which has the instructions for how to make the proteins that would be the antigens for the virus. So basically they take the virus and they find some particular proteins on the virus, and then they like back engineer how they would be made, and then they inject the instructions into you, and then your cells make the the antigens, and then your body responds to that. So it's a much safer kind of vaccine because you're not actually injecting any virus at all. You're just injecting instructions for how to make a very specific protein that the virus contains, and then the body reacts to that. Does that make sense? Sorry, it took me a while to get around to it, but that's kind of the idea of how mRNA vaccines work. And then Matt, you said, so probability of having a side effects are 1 out of how many? If it's 2 to 4%, well, it'd be 2 out of 100. Right, so the way percents work is 1 out of 100 is 1%, right? So if you have 2 to 4%, you're talking about 2 out of 100 people, which is, you know, that's a lot of people because this is going to be administered to millions of people. So you're talking about, you know, tens of thousands of people. But the side effects are mild. So, but if you look at like a normal flu vaccine, let's see, percent of people who have side effects from flu vaccine. Okay, according to the CDC, mild side effects from the flu shot, 1 to 2%. So, okay, so it's like double. It's double. But yeah, like Alexi said, it's way better to have like a slight headache, to have like a 2 to 4% chance of getting a slight headache and some fatigue than having the actual virus. So the side effects, you know, a little bit high, 1 to 2, like compared to a flu vaccine, but you're never going to get out of a situation of having any side effects at all. That's a good question, Jeremiah. I'm not sure how like I was looking at this article. Let's see, what does it say about the side effects? Where is this? Here are the side effects. It doesn't say how long they last, but, you know, typically the side effects only last for a couple days. You know, you like in my experience, you know, getting the flu shot, I've never had terrible side effects from the flu shot, but you know, you get the flu shot and you have a little bit of pain, you know, in your shoulder from that on the site that it's injected in for like a day. And that's totally fine. I don't know. I don't imagine them lasting longer than like a week at most, probably, these side effects. But I don't know. It doesn't say in this article, at least. Okay, so I think that's pretty much all we need here about the first one. So that's Pfizer's mRNA vaccine. Let's take a look at, what's the next one? Novavax's. What's theirs look like? November 3rd. November 11th. Novavax announces COVID-19 clinical development progress. Novavax had 15,000 people. They had some trials in South Africa, some in Mexico and the United States. I don't like, that's not a good article. Let's see. Let's look at Science Mag. Will a small, long-shot U.S. company end up producing the best coronavirus vaccine? Okay, yeah, so if you look at the list of companies, like Pfizer, huge. AstraZeneca, huge. Moderna, huge. I don't know Janssen's and I don't know Novavax, so I'm assuming these are smaller companies. Let's see. Oh wow, this company was delisted by the Nasdaq. They had a failed vaccine, 2 failed vaccines in 3 years. Their shares plunged to less than $1. Wow, they were in some dire straits. Novavax is slated to receive $2 billion from the government. Let's see, what is their vaccine? Where's the info here? Okay. Here we go. Okay. I'll move my camera over here so we can see a little better. What's up, Shanna? Do I consider SLO to be a spaced out campus? Do I consider SLO to be a spaced out campus? Spaced out, like, you mean like spread out? Like physically spread apart? Do I consider it to be spaced out? It's not like, I wouldn't say it's massively spaced out. Like, and it's changed. You know, I went there. I was there. You know, what is it? 10 years ago. So they could have had a lot of construction in the meantime. But as far as I remember, and I was in the science department, so it was like, felt pretty normal, like not incredibly compact, but like decently spaced out. I felt like it was pretty good. But it's hard to say because you have to like compare it to other campuses. And I only ever went to SLO. And it's definitely more spaced out than like Monte Vista, for sure. And it's probably more spaced out than a university that you would find in a urban environment like UCLA or USC or maybe even UCSB, because San Luis Obispo is more suburban-y, more rural, has a lot of open space. So I would say if you're trying to pick a campus that's spaced out in California, that might be one. Another one that I think is probably pretty spaced out might be UC Merced. Because if you look at UC Merced, that's like the newest UC, right? And it's built over in like, not to be rude, but kind of the middle of nowhere, right? It's way over here in Central California. And it's kind of like, out in the middle of nowhere, to be honest. So that would be spread out probably. I don't know though how the architecture of it. But yeah, I mean, all things considered, I would say San Luis Obispo's campus is pretty spread out. And you can kind of see, like, you see, like, are you talking about like you can kind of see, so when I went to Cal Poly, I lived, where was I? I didn't live close to campus. The nice thing about Cal Poly is that you can kind of live pretty far from campus and it's still not too bad to get there. The traffic isn't ever too bad. But, so you can spread yourself out in terms of where you live. Like I lived back, here's Foothill. Oh, I lived way over here, didn't I? No, where did I live? Oh, back here. Wait, oh my God, I'm so messed up right now. South Street, Broad Street, Orcutt, Tank Farm. Okay, I was in over here. Here we go. I lived like over here. Whoa, wow. Actually, I lived way further away from campus than I remember. I lived like in this spot. And I motorcycled to school. I had a motorcycle and I would just cruise in this way. But the campus itself is pretty decently spread out. These are all the dorms over here. This is like their new dorms. I stayed in like these old red brick ones when I was there. This is like the science area here. This is all science. Engineering, I think, is over here. Business was down here. Athletic stuff is all over here. Yeah, it's decent. It's decently spread out, but not like massively. Not massively. And I don't know if you should pick your college just simply based off of whether it's spread out or not, because that's only really going to matter potentially for like 1 year, not even. Like if the vaccine comes out, like, unless you just like having things spread out anyway. All right, let's take a look at what Novavax is. So, to make their vaccine, Novavax scientists first used a baculovirus to insert the gene for SARS-CoV-2 spike protein. Okay, so this spike protein is the same protein, as I'm guessing, that Pfizer back engineered to make their mRNA vaccine with. So the mRNA that Pfizer made codes for this spike protein, and it looks like Novavax hold on, let's read this further actually. So Novavax scientists first used a baculovirus, okay, which is a different virus, to insert the gene. So they use the gene, so they use the DNA instructions for the spike protein into moth cells, which produce the spikes on their membranes. Scientists then harvested the spike proteins and mixed them with a synthetic soap-like particle in which the spikes embed. A compound derived from trees serves as an immune-boosting adjuvant. Remember yesterday we looked up the word adjuvant? Adjuvant means increases the effectiveness of the vaccine. So oftentimes vaccines are administered with an adjuvant, or medicines are administered with an adjuvant to help increase their effectiveness. So let's take a look. So the baculovirus delivers the spike protein DNA from the coronavirus to a fall armyworm moth cell. Okay, so boom, this virus injects the DNA, this moth cell makes the spike protein, and now the spike protein is on the surface of the moth cell. Then they harvest that spike protein, mix it with the saponin, a purified plant compound, is the adjuvant. Oh, you're right, Alexa. Yeah. Sorry. I shouldn't give Pfizer all the credit. They're just like the big guys. So I should say this isn't Pfizer's mRNA. This is, what was it? It was BioNTech. Yeah. Good call. Okay, so it looks like then they're literally injecting the actual spike protein. As a 34-year-old graduate student, hey, this person's exactly my age, graduate student at Texas University he was 34 in 1983, so he's old. Okay, whatever. With colleagues, had developed a system that could produce proteins to be quiet. You should start it. Okay. Let's keep going. I don't care about their shares. Well, okay. So here we go. Oh. Novavax's, what would we call this? Novavax's spike protein antigen vaccine. That's kind of the way I'll describe it. All right, so we know that it is a use moth cells to generate the spike protein that is found on the virus. The spike protein is then harvested and combined with an adjuvant and injected to elicit an immune response in the patient. So basically we can say uses actual virus proteins, which is different than the mRNA one. Is there any data on their effectiveness / their side effects and stuff? There might not be any information out yet on this one. Let's look at this paper. Phase 1 to 2 trial. Oh, this was the phase 1 to 2 trial. Results. Okay, after randomization, 83 participants were assigned to receive the vaccine with adjuvant and 25 without. And 23 were given a placebo. Okay, so they were comparing, they had 2 experimental groups. They had their control group, which got the placebo. And then they had their 2 other experimental groups. They had one that received the vaccine without the adjuvant and one that received it with the adjuvant. So they probably wanted to see, like, how effective was the adjuvant at increasing immune response. No serious adverse events were noted. Reactogenicity was absent. Reactogenicity, holy. That must just mean like how much a person reacted. Reactogenicity refers to a subset of reactions that occur soon after vaccination and are a physical manifestation of the inflammatory response to vaccine. In clinical trials, information on expected signs and symptoms after vaccine is actively sought. Adverse events that are common. So it's not really, it's like kind of like side effects. Kind of, I guess, like side effects. I guess we'll just sort of. But these were in their phase 1 and 2 trials. This is a little early. I'm kind of, because they're apparently in phase 3. You don't really want to go to the actual company's website to get information because they're like, obviously they have incentive to like not show some things that are bad. But I want to see, okay, pivotal phase 3 trial in the UK completes enrollment. Completed enrollment, but it hasn't been completed. Wait, was the Pfizer data that we were looking at, was that, hold on. Maybe all of these phase 3 trials, the data's not out yet. Maybe the Pfizer one that we were looking at was actually data from phase 2. I just got to go back. I'm trying to find where that tab was. No. This is the one we were looking at. Oh no, it was phase 3. It was phase 3. So this was phase 3 data here. So this was the BioNTech's is phase 3 data. This is phase 3 data. It looks like we might only have phase 2B data for Novavaxes at this point. All right. I did not know that. I don't know much about what Russia's doing. What do I think of Russia releasing their vaccine back in August before phase 3? What's up, Ahana? How are you? Let's see. So what did they do? They just said, we're not going to deal with this, and we're just going to... Which vaccine did they release? They actually started vaccinations already? Sputnik 5. They can't give up on the name Sputnik. The Russians love Sputnik. They're like, you know what? That was the name of our, spaceship that was the first in space or whatever it was. Everything will be Sputnik. Nice. Thanks for dropping by, Ahana. Always nice to see you. Let me know if you have any questions. I'm just doing a little research on vaccines. They finished phase 2 testing, and one of the participants was Putin's daughter. So they pushed it out before phase 3 to vaccinate healthcare workers. Well, I mean, they I guess they technically do whatever they want. It's their country. They get to decide. They have their own regulations, I'm assuming. Feels like this is the space race all over again from the 50s and 60s. Every country's like, we must be first. In a move criticized by many scientists as reckless, Russian regulators licensed the vaccines for limited rollout in August without waiting for safety or efficacy data from phase 3 trial. The vaccine is composed of 2 different adenoviruses that produce the coronavirus spike protein, administers 3 weeks apart. Okay, so they did something maybe similar to what the Novavax is trying to do. It's not an mRNA virus, or vaccine rather. No problem, Mahana. The grade calculators are always nice. They're a little handy just to give you some info. Well, Russia's fast-track coronavirus vaccine draws outrage over safety. I mean, we'll find out, right? I mean, that's the risk. If you go early, hey, you could get away with it. You could be lucky, but it would not be good for Russia if it turns out that they have some crazy side effect that they didn't catch. It is a risky decision. And the question is, why did they decide to do that so quickly? I mean, for obvious reasons, just because one, it could be they just want to be first, right? So that would be the worst reason to do it, is just so that they could be like, we were the first ones. Now, if they did it, but the other reason could be they were doing it just because they want to get this under wraps quickly. But still, though. This is a reckless and foolish decision. Mass vaccination with an improperly tested vaccine is unethical. Any problem with the Russian vaccination campaign would be disastrous, both through its negative effects on health, but also because it would further set back the acceptance of vaccines in the population. That's true, right? So if Russia goes and administers this vaccine and then it causes all these side effects, all these anti-vaxxers now have more, more quote unquote evidence to prop up their beliefs that, obviously this wouldn't be evidence that vaccines don't work. All this would be is evidence that vaccines that are not properly vetted don't work. Yeah. In his announcements, Putin said that the Russian regulator had approved a COVID-19 vaccine developed by Gamaleya Research Institute, even though phase 3 trials had yet to be completed. The vaccine was given to 76 volunteers? That's it? Just 76? Wow, they really... Because that is not a good enough sample size. 76? Because Pfizer's is doing what? They had 42,000 people in the trial? Yeah, well, sounds pretty sketch, pretty sus. I just got to hope that nothing comes out of it. Yeah, well, I guess the only thing is we just have to hope that it doesn't go bad for them. All right, let's take a look here. Let's look at, we got to finish up with our Novavax one. So looks like they're still in we don't have United Kingdom pivotal phase 3 trial update. So they enrolled 15,000 people for their phase 3 trials. But interim data in this event-driven trial are expected as soon as first quarter 21. So let's put that down. Phase 3. Let's put down phase 3 trial data will be available early first quarter 2021. Okay. Do we have any data from this on their phase 2 trials? All right, well, it doesn't look like it, so I'm just going to go ahead and go. Okay, next, Moderna's. What is Moderna's? All right, New York Times. You came out today, fresh, hot off the press. Hot off the press. Oof, I feel the heat coming from my monitor with how hot this is. All right. Moderna, a Massachusetts-based vaccine developer, partnered with the NIH to develop and test a coronavirus vaccine known as mRNA. Ooh. Another mRNA vaccine, huh? A clinical trial demonstrated that the vaccine has an efficacy rate of 94.1%. What trial? What phase? Okay. All right, Russia. Go ahead and claim that. Like, let's see what they're... This is just referring to the... how many people did they have? Is this the same one that had like 94 people? Or like... Yeah, 20 people. Okay. All right, you can't... 20 people is not enough. What about the Moderna? How many people did they have? All right, early data shows Moderna's coronavirus vaccine is 94.5% effective. How many people? Wait, where's the info on this? Was 94% effective. Where is the info though? Like the actual information. Okay. Let's see. Ahana, chromatids are copied to form blank before meiosis 1. That's okay. It's okay, Ahana. This is a little bit of a science stream anyway today. I was hoping to play Minecraft, but I got to get through these 5 vaccines. Then we'll play some Minecraft. Let's see. Sorry. Wait, did I mean chromosomes? So, look, if you do, let's see, chromatid versus chromosome. Just to show you. So, the word chromosome and chromatid are slightly different, right? So, a chromosome is the entire thing. When it's duplicated, you have 2 chromatids. So there is a difference. Yes, the chromosomes are... Oh, I see what you're saying. Yeah, chromosomes are copied to form... what would it be? Chromosomes, this is... Wait, let me see. Before meiosis 1. So this is before the process even starts. Chromosomes are copied to form sister chromatids is technically what it would be, if that question is correct. Yeah, so it would be chromosomes right here. So this is a good picture to show that, right? So the replication is what's happening before meiosis 1. So you have your chromosomes, which are replicated to form sister chromatids, which are the copies of each other. I'll be right back. I got to use the restroom. Yes, Ahana, that's correct. That is correct. All right. So it looks like Moderna has an mRNA vaccine as well. How is theirs different than the BioNTech's mRNA vaccine? Parsa. What's up, my memer? Let's see here. So Moderna's mRNA vaccine, what's the difference, right? How are they different? Okay, well, that's interesting. Needs to be refrigerated and stored at -4. Was that Celsius? No, F. Fahrenheit. Should be available and should be stable for up to 6 months when shipped. Needs to be refrigerated and stored at -4. Can last, what was it, 6 months? Let's say 6 months. Yeah, Ashi, we're definitely debrief it. Meiosis is a very confusing process, so I'm definitely going to take time to go through the process and sort of explain it as best I can from my perspective and answer questions. So I will definitely be debriefing it. But if you have any questions now, feel free to ask. Parsa, are you all done with your college apps? That's pretty legit, if so. Okay, so boom, there's a little thing. mRNA goes into your cell. It's translated, creates the protein. The protein is displayed. Oh, wait, here, this is where it gets into some of the MHC restriction stuff, actually. So it looks like, based off this diagram from our friends at the New York Times, some of the spike protein is presented by MHCs, which are these antigen-presenting things that stick the antigen this off, and then sometimes they just stick through the cell's surface. Some of the spike proteins form spikes that migrate to the surface of the cell and stick out their tips. The vaccinated cells also break up some of the protein into fragments, which they present. The protruding spikes and protein fragments can be recognized. When a vaccinated cell dies, the debris will contain many spike proteins and protein fragments, which can then be taken up by a type of immune cell called antigen-presenting cell. The cell presents fragments of the spike protein on its surface. When other cells called helper T cells detect these fragments, the helper T cells can raise the alarm and help marshal other immune cells to fight. This is just what happens after the vaccine. This is all stuff we know. Yep. That the B cells produce the antibodies. Yep. This is probably a really good article for those of you that want to get a little bit of background on just generally how the thing works, how vaccines work in general. All right. Okay, here it is. After initial studies yielded promising results, Moderna began phase 3 testing, 30,000. In July. Wow, their phase 3 started in July. That's really early. Do we have results then yet? Oh, we already looked at this article. Okay, I already looked at both these articles. They don't have any of the data. Nice. Parsa, glad to hear that you'll be all wrapped up before the end of the year. That'll be nice. Put 2020 behind you. Put a bow on it and send it. Spring 2021, vaccines by Moderna and Pfizer are expected to reach large-scale distribution in the spring. Yeah, we're not coming back for school in the second semester. It's just not happening. It's possible. Yeah, no, never mind. God, New York Times, do you only link to your own articles? Come on, man. Are all these? Watch this. Look at this. New York Times. Do you have any outside sources in your articles? Promising results? Oh, there you go. Thank you. Something outside of the New England Journal of Medicine. Okay. Preliminary report. Here's what I was looking at. We conducted phase 1. Oh, this is phase 1 though. Where do we find... Okay, I already did that one. So basically we can say Moderna is basically the same as BioNTech's, right? In that it requires 2 shots, approximately 28 days apart. So I guess Moderna and BioNTech are basically developing the same exact technique of vaccine. This mRNA vaccine. An early study found that Moderna's vaccine provides protection for at least 3 months. Okay. Okay, well, I can't. I'm having a hard time finding the actual preliminary data from phase 3 because all I get is links to other New York Times articles, and then within those other New York Times articles, I click on this one, and it takes me to another New York Times article. Does this one have the data? No data. What about this link? Where does this go? This takes me just to a random article. So the problem is the New York Times. This is just bad. You need to have a link. If you're going to state something, this is just a little tip for all you guys out there writing. When you write things, and you need to cite your sources, you should have a link where, so it says, announced that it was 94.5% effective. Do you have a link down here? Maybe at the bottom? Where are your sources? Nothing, huh? Nothing. What's up, Haxwell? Vaccine isn't permanent, so I'm doing some research right now. The BioNTech and the Mrna Vaccine that are in phase 3 right now, both of these are at least 3 months effective. And what I read earlier was that, what did I read? Wait, did I not record that somewhere? Oh, here. Right now, they're not 100% sure how long the immunity lasts. And they're saying someone from the BioNTech stuff said at worst a yearly booster, so it'd be kind of like the flu. Yeah, exactly, Ashi. So like that's kind of what would be expected at worst, is that it's not a permanent vaccine, that it's a yearly vaccine, maybe. I'm really shocked that the New York Times does not have a single link to the actual data. That they report this 94.5% effective figure, but I'm not getting anything. But I want to know, like with the BioNTech, we saw the data was, a certain number of people. They had 170 people that got infected, and 95% of them, but only 8 of them had the, wait, what was it? Only 8 of them, 8 of the people that got infected of the 170 had the vaccine. So it was at 95% effective, because 8 out of 170 is 95, I think. Wait, is that right? Does that make sense? Yeah. Let's see if we can just look it up. Promising interim results from a clinical trial of Mrna COVID vaccine. Let's see, what do we got here? Here we go. Here we go. Found it. Okay, so the findings are statistically significant, meaning they are likely not due to chance. 90 of the cases occurred in the placebo group. 5 occurred in the vaccinated group. Okay, so this is N equals 95. Okay, N equals 95. Well, this was N equals 170. So the BioNTechs had a higher sample size and a slightly higher effective rate, but both are really good. That's really good. So I think that's all right. All right. Anything else we should know? Side effects? Probably pretty similar. This is the Mrna website. Wait. Hold on a sec. What does this say? Today announced the preliminary efficacy of the phase 3. 196 cases confirms the high efficacy. The data analysis indicates efficacy of 94.1. Oh, here it is. Yeah, 95 cases. Oh, these are updated. This is an updated number. 190. Efficacy is 94.1. And the N equals 196. We got updated numbers, everybody. Look at that. Fresh numbers. 196 cases. The most common adverse reactions included injection site pain, fatigue, myalgia, arthralgia, headache, arrhythmia, Okay. Okay, cool. All right, Moderna's done. What do we got? Janssen's and then AstraZeneca's. What's Janssen's? Let's see. Oh, Janssen is Johnson & Johnson? Janssen & Janssen? Wait, let me guess. Janssen is the producer and Johnson & Johnson is the distributor? Oh, Janssen is a subsidiary of Johnson & Johnson. Okay, we got 60,000 participants in their phase 3. In addition to a single dose regimen, Janssen has now initiated a 2-dose regimen. Okay. Johnson & Johnson, Aussie, is a huge company. Johnson & Johnson is like massive. They have pharmaceutical, they have like all these things underneath them. They've got like, yes, they do make things like you said, creams and like, you know, the stuff you would find, you know, in the bathroom of like a household, like cleaning products. But if you look at Johnson & Johnson's, like, they are a monster company. Like, they are, let's just go to their Wikipedia. Yeah. $85 billion company. That's how much their assets, $245 billion. Their subsidiaries, huge numbers. We call this a multinational corporation. their products, right? They have a lot of pharmaceuticals. They have neuroscience, infectious disease, immunology. They make stuff they make, like they have companies that produce drugs for them for different medical issues. They produce different medical devices like what is this? Aesthetics? Okay. Stuff for diabetes, ear, nose, and throat. And then this is what you're thinking about, Ashi. Baby care, nutritionals, oral health, skin and hair care, right? So this is like their consumer. This is all, like, as a consumer, this is what you see from Johnson & Johnson because this is what's on the shelves. But they have all this other like really high-level medical devices and drugs too that you don't see because you don't go and do that stuff. It's out of your world. All right, Janssen's vaccine. They're testing a single and a double dose. Dose. 4 days ago. Johnson & Johnson starts rolling review for COVID-19 vaccine in Europe. All right, what is this one? The Janssen COVID-19 vaccine is a recombinant vector vaccine using a human adenovirus to express the SARS-CoV-2 spike protein in cells. It uses the company's AdVac vaccine platform, which was used to develop and manufacture Janssen's Ebola vaccine regimen, approved by European Commission and constructed Zika. Oops. I want to see some of the data on this. Wait, is this another mRNA vaccine? Because it says when a person is given the vaccine, their cells will read the genetic instructions and produce the protein, the spike protein. The person's immune system will then treat this protein as foreign and produce antibodies and T cells against it. If the vaccinated person comes into contact with SARS-CoV-2, the immune system will recognize the virus and prepare to attack it. Basically the same thing. So it looks like Janssen's it could be a single dose, which is a perk compared to the other two big ones. It seems like, based on so far this research, like it seems like Novavax's is like kind of like trailing off. Like it doesn't have as much phase 3 trial data as like Moderna and Pfizer's. It's also not an mRNA vaccine, which kind of makes it like less good. Doesn't seem like Novavax is going to be the one. Like it seems to me like it's a race between Pfizer and BioNTech's and Moderna's right now. And maybe J&J might have an advantage because it's only a one dose vaccine. I still haven't gotten enough information yet about how this works, if it's an mRNA or not. This is old. Oh, this is just their update page. Didn't say anything about what kind it is. How many homologous chromosomes do humans have? We have 46. Because you have 2 homologous chromosomes per set, and you have 23. So it's 23 times 2. We have 23 different chromosomes. And for each of those, you have a maternal and a paternal chromosome, which are homologous to each other. So it would be 46. Yeah, or 23 pairs. That's also totally fine to say. They mean the same thing. Okay, but here we go. The two vaccines work in very different ways. The Mrna vaccine delivers like mRNA. Okay, good. The cells use the vaccine to produce the protein. In the Mrna study, researchers vaccinated monkeys. The Johnson & Johnson vaccine, in contrast, is based on a virus called 8026. Okay, based on a virus called 8026, which researchers have modified based on a virus called 8026, is modified to carry the spike protein gene. and slip into the cell, into human cells. The cells then basically do what they do, and they make the proteins. Then use the gene to make the protein. So what's the difference? So the difference is basically the method of the method in which the gene gets inside the cell. How many sister chromatids do we have? It depends. You have 0 before DNA is replicated, right? So sister chromatids only exist after DNA has been replicated and before the cell divides, right? There's like a period of time where the cell has like too many chromosomes because it's going to split and separate them. So during that period of time, you would have each homologous pair would be doubled. So you would have 46 times 2? You'd have 92. Yeah, 92. For a short period of time, you'd have 92. And then they would be split. Yep, exactly, Ashi. Ashi, you're on top of it. Ashi, I'm impressed. You know just as much as I do about that. Good stuff. Okay, so seems like the difference this is different from the mRNA vaccines of Moderna and Pfizer because it uses a virus to enter the cell rather than a lipid nanoparticle. Let's see, are there any preliminary phase 3 results? Preliminary phase 3 results from Janssen. I don't think so. I don't think so. First participants were dosed in the phase 3 trial on September 23rd. So that was a little while ago. Announced the launch of its large-scale phase 3. So it sounds to me like we have, of these 5 vaccines that are in phase 3, so far, I've looked at 4 of them. I haven't looked at the Astrazeneca one yet, but it seems like Moderna and Pfizer have preliminary results from their phase 3 trial, whereas Novavax and Janssen do not yet. Okay. Let's see what our final candidate is like here. So this is going to be our Astrazeneca vaccine. Janssen & Janssen's. What should I call this one? Janssen & Janssen's. I'm just going to call it Janssen & Janssen from now on. Janssen & Janssen's. This would be like, I would say, viral vector based. Viral genetic vector based vaccine. We'll just say that. Genetic viral vector based vaccine, because it's using, yeah, that's fine. Nathan, that would be so here's the thing, like, this is the only way you can remember stuff. Like, so when if you're like studying and you're trying to like remember material, like the only way that really works is if you, as you're going through the material, like you translate stuff into your own words. So like as I'm reading through all of these articles, like I'm not just reading them, I'm like processing them in my brain and going like, how can I make it make sense to me? And then I'm writing things down in my own words as like summaries of like important things. And that just helps me. Okay, we're almost done. We can almost finish these, and then I can play some Minecraft and get relax for a little bit. Almost done with some research here. Dang, I've almost spent 2 hours so far on this. All right. Astrazeneca, here we come. All right. What to know about AstraZeneca's vaccine controversy? Spicy. On December 4th. AstraZeneca announced that its vaccine is 70% effective. That is not great, considering that these other ones are in the 90s. But a major dosing error during the trials may have affected the overall efficacy. Some clinical trial participants were mistakenly given half dose rather than a full dose. Ooh. Researchers discovered that those who were given the weaker doses produced a better immune response? Weird. So like they inadvertently messed up the dosage. But yet it actually seems to be better? All right, let's see. Okay. AstraZeneca's COVID-19 vaccine is thought to be one of the biggest game changers when it comes to getting a handle on the pandemic. Unlike some other vaccines, AstraZeneca's vaccine, which is given in 2 doses. Okay. Let's get this down. 2 doses a month apart, as usual. That's pretty standard. Although I remember the other one. What was it? This one currently is going to be a single dose, but they are researching a potential double dose that would be like, what was it, like, I don't know, something like 60 days apart, or no, 80. I think it said like 86 days apart, which is crazy. That's really long. 3 times as long as normal. What's up, Zenda? How goes it on this Saturday afternoon? So 2 doses a month apart, pretty standard. And what did it say here? The upside to the AstraZeneca one is it does not need to be stored in super cold temperatures. So we can say does not need to be stored in super, but I don't know what that means. Super cold temperatures? Like, what is super cold? Like, come on, that's very vague. Each dose is significantly cheaper than the other shots and will be easier to distribute. Okay. So it does not need to be stored in super cold and is cheaper per dose. So the AstraZeneca one seems to have some advantages here. Crawling your way to second semester senior. Almost there, dude. You got what? Like a month left at most, maybe 3 weeks. Good question, Zenda. That is what this document is all about. There's five vaccines that are in phase 3 trials right now, and I'm going through and I'm researching all 5, and I'm making a short bullet point list of what each one is. So here, I will share this doc with you guys. If you're interested in seeing my research, go ahead and click on that so far. Okay, AstraZeneca's vaccine. I'm on the last vaccine right now. 2 doses a month apart, pretty standard. Does not need to be stored in super cold temperatures and is cheaper per dose, which is an upside compared to Pfizer's and Moderna's. 2 doses a month apart, pretty standard. Let's see. These were these phase 3? When was this article? December 4th. These must be phase 3 results. This is very interesting. So when regulators informed us that nearly 3,000 participants had received a lower dose, they permitted the trial to continue as the dosing error had no impact on the vaccine's safety. In fact, the dosing mistake led to an interesting discovery. In the participants who received 2 full doses, the vaccine efficacy was 62%, but those who received a half dose and a full dose, it was close to 90. Wow, crazy. So let's put that down. Preliminary phase 3. So J&J, no phase 3 data yet. No phase 3 trial data yet. And then these ones we have phase 3. So it's a preliminary phase 3 data. And then same thing for Pfizer's. And for AstraZeneca's, we've got some preliminary phase 3 as well. Preliminary phase 3 data had an error that revealed I'll just copy and paste this in there. Okay. So overall, preliminary results suggest that it is 70% effective, but we could say but is more likely to be approximately 90% effective given this new dose schedule, right? That's like half dose, then full dose. Some experts have criticized the fact that AstraZeneca combined the efficacy results from what's essentially two different trials. Okay, that's fair. And say the company will need to conduct another trial properly evaluating the effectiveness of the half-dose, full-dose regimen. Some small mistakes are common, but giving thousands of participants the wrong dose unintentionally is not a common mistake. Time will tell whether this particular mistake leads to a discovery, but at this point in time, there's a lot of uncertainty around the findings. Smith, whose research focuses on public and health safety, believes AstraZeneca will seek approval for the full dose, which is 62% effective. The big question is whether regulators will trust the 62%, considering the sample size was smaller. So what was the sample size? Did they have that in here? 3,000. So this was 3,000 participants. Oh, wait. 3,000 received the lower dose. So the sample size was larger than that. Got to find some more articles here. Let's see. Oh, wait. Oops. That's Janssen's. I don't care about Janssen's anymore. All right. Let's take a look at New York Times. Trusty old NYT. Are positive but confusing, yeah. This month has seen a torrent of news about experimental vaccines. Preliminary analysis showed the vaccine was effective, especially when the first dose was mistakenly cut in half. Zenda, what are you trying to do in the future? Are you trying to get into pharmaceuticals? Nice. That's a very it's a tricky industry though. It takes a long time. Like what part of it would you be interested in? Are you looking at like the research side, the like production side, right? It's like big industry that has a lot of different aspects to it. All right. What is the AstraZeneca vaccine? Have I got that yet? No. Researchers at the use kind of virus called adenovirus. It's because of the cold. It's generally altered virus that carried a gene. Okay, so basically the same thing as the Johnson & Johnson one. So we'll say this is we'll say like very similar to the J&J slash Janssen's vaccine in that it uses a modified adenovirus to insert genetic information into the host cells that then use the gene to make the spike protein. Okay. Yeah, of course, Ahana. Take these notes as free. These are all yours to do what you will with. And this is the one that this is the same one that Russia is using. So maybe we'll put this down here. Let's do this. We'll do this. Russia's Sputnik 5 vaccine. Uses the same adenovirus technique that the what is it? So we've got 3 that use this now. AstraZeneca, Pfizer, Oh no, not Pfizer. Sorry, Pfizer's the mRNA, right? Yeah, no. So it's Johnson and Johnson. Wait, AstraZeneca. And Johnson and Johnson. Okay. I mean, yeah, I guess that's kind of what college is for. But the thing is, like, depending on where you go to college, you sometimes have to declare your major ahead of time, and the major that you would go for could vary wildly depending on like what you're interested in doing in a pharmaceutical realm, right? Like, if you want to get into research, you might need to major in like biochemistry, but if you're interested more in like I don't know, like the other aspects of it, I guess. Well, I mean, I guess if you're going to get into biotech stuff, you'd probably just be more interested in the actual research, which would be biochemistry and biotechnology. But I imagine like there's the whole business side of it, right? Which, you know, is a whole nother area. Bye, Ashi. Or there's even like the engineering side, like the machineries that's used for it. Okay, so you're kind of leaving it open. That's pretty good. Uses the same adenovirus technique that AstraZeneca and J&J are using. But the thing is, they did not have any phase 3 trials before they administered it to the general population. Okay. So just some interesting things here, maybe to put at the bottom, actually. Hold on. Let's just finish up. I'm not done with this. In the spring, AstraZeneca and Oxford started running clinical trials. Okay. Okay, 131. So wait. That's fine. How does it stack up against other candidates? Josh, I am Aquaman from the hit TV series The Boys. Why did you say that like you were writing a magazine article? Aqua from the hit TV series The Boys. Who is that? Aquaman from The Boys. Yeah, that's me. Yep, you got it. Yeah, that's me. What's up, man? How you doing? I'm just chilling. You know, we're not filming anymore because of COVID. I got my suit still. It's hanging in my closet. Sometimes I'll just wear it at night and just kind of hang out, just relax, you know. I often stand like this pretty regularly and look at myself in the mirror. That's just fun. You know, I actually haven't seen this. I've actually seen like 2 episodes of this show, and I think the problem was like I just couldn't get into it. It felt a little bit. I'm a very like I prefer shows that are very not so over the top, which is why I often have a hard time getting into like superhero movies. You know, like the whole Avengers series was one that missed me. How does AstraZeneca stack up? The only thing that you can really say right now is that the vaccine seems to work. It's just hard to say how well it works compared to others. Experts have a hard time parsing the results because of the way they were announced. Right, because of the issue, yeah, with the half full dose thing. Like the results from Pfizer and Moderna, the data was summarized in a news release. Here we go. This is see, beautiful. Thank you, AstraZeneca. You provide the actual information, the numbers. That's what I want. So we could say N equals 8,895, which is a pretty big sample size. The biggest that we've seen so far of any of these. N equals 8,095, and then for the 90%, it was N equals 2,074 or 2,741, which is actually pretty good too. Okay. Okay. Whew. All right. After 2 hours of research, we have at least a little bit of a summary of the 5 potential vaccines. Now, let's anytime you finish doing some research and you finish sort of taking notes on something that you're learning about, a good thing to do is what I call a final summary. So what I would do is I would go down here to the bottom, right? And I would go like, okay, what are the takeaways? So we go summary / takeaways. I would say like, let me see here. As of December 5th, we have 5 potential candidate vaccines in phase 3 trials. Two use mRNA techniques, which have never been approved, which have never been approved by the FDA before, so would be the first of their kind. They are advantageous because they are not MHC restricted, and they do not require any viral particles to be injected. Right? I think those are the big advantages. One uses a relatively older technique of injecting. This was the Novavax one. What does it do? The Novavax one? Right, the spike This is like uses actual virus proteins. This is sort of like your old illicit. This is a way to this is the older style. Two use mRNA techniques. So that would be Pfizer and Moderna, right? Yeah. One, Novavax, uses a relatively older technique of injecting actual viral spike proteins. And two, use, this would be J and J and Astrazeneca. Use a so this uses an adenovirus technique to inject genetic material this one is like older. This has been around, but only I think one vaccine exists that uses the technique. And it's for Ebola. Okay. The leading candidates so far seem to be, I'm going to say the Pfizer and Moderna, seem to be Pfizer and Moderna as they have released some preliminary data with very good results. However, AstraZeneca has some advantages that may make it a more viable option for distribution. Those being it is cheaper and does not require cold temps to store. But then again, does the Janssen & Janssen one need to be starred? If it's using the same technique as the AstraZeneca one? Maybe. Okay. All right, I'm happy with that. I think I got it. So, pretty good. Let's do a little Minecraft. Chill it out now, huh? Oh, where did my my video disappeared? The heck? I lost my video. Just disappeared. What the heck? Wait, what the heck? Video capture, display capture, group 2, image. Oh, that's right. That should be in there. What the heck? This is so weird. All right, I'm going to have to just do a quick restart of the stream, I think, because I don't know what happened. I'll be back in 2 seconds. 2 seconds.